Hi again. On this lecture, which is our third lecture in Module 7, our third of 4 total, I want to go over atrial natriuretic peptide, ANP, aldosterone, and RAAS again. And a lot of this should be review, but I know that to learn this material you have to review it numerous times. The old adage for how the brain works is that you have to repeat to remember. So you have to repeat things a number of times. So I don't mind doing that. That's how people learn. That's how I learn. So let's go ahead and start with the first slide. And I just found these slides. I really liked them. Rather than me drawing them, I figured, well, I'll just kind of walk you through this. But some of this we've already been through, but these are nice summary slides. They're nice and clear. So this slide is about ANP. So if you want to go and review what ANP is all about, this is a great slide. And that, of course, stands for atrial, referring to the atria of the heart, natriuretic, referring to natrium or sodium. That's where the Na in sodium comes from. Natriuretic peptides, or peptide. Turns out there's a whole family of these things. Okay, so that's what this picture is about. And what ANP does is that it promotes excretion of salt, primarily sodium and water. So we're excreting it, we're not retaining it, we're getting rid of it. So to start, increased blood volume at the top causes increased atrial stretch. So we've already talked about that. We did that back in the cardiovascular system. So those atrial cells, that when they are stretched, they release ANP. And this is our ANP. Remember what we're trying to do. Don't lose track of the goal. It's to get rid of salt, and water follows salt. So we're going to dump them both. So ANP has a number of targets shown here. And here are 4 targets for that hormone at that level. And you should be able to work through these responses. You could make flashcards for some of this. You could have on one side have targets of ANP. And on the back side of the flashcard, you'd have hypothalamus, kidney, adrenal cortex, medulla oblongata, etc. You could make another flashcard that asked you to explain how the hypothalamus is infected by ANP to reduce the salt concentration. So you should practice working through these responses. Let's take a couple of them. Okay, so here's the ANP. It's released. We have high blood volume, goes to the hypothalamus. So there are ANP receptors on the hypothalamus that bind ANP, and that causes the hypothalamus to release less vasopressin. Well, if we have less vasopressin, we're going to be reabsorbing less water. Good. That gets us to more water excreted, less ADH. So less ADH equals more water excreted. There are receptors for ANP on the kidneys. So it increases GFR, decreases renin. So if we increase the GFR, we dilate the afferent arteriole, the GFR goes up, and that also results in increased loss of sodium and increased loss of water. We just filter more of it passes through into the bladder. And the kidneys can also affect the sodium reabsorption, and under the influence of ANP they would absorb less, so that more sodium would be excreted in the urine. The decreased renin leads to blood pressure. Let's skip over to the adrenal cortex. ANP targets the cortex. Remember, what comes out of the cortex of relevance here in terms of sodium is aldosterone. So we release less aldosterone, we absorb less sodium, etc. And the medulla oblongata is affected by ANP, lowers blood pressure. So that would be through the cardiovascular control center. So if you could list those targets, hypothalamus, kidney, cortex, medulla, etc., and say something about what they do, that would be great. So hypothalamus, less ADH, less water reabsorbed, etc. Okay, so that's the slide for ANP. This is pretty much it. If you remember this much, you've got it for that hormone. And we've already done ADH, which we just did. So let's talk about aldosterone again. So some key points to not forget is that the connection between sodium and the ECF volume. More sodium retained, more fluid retained, increase in blood volume, increase in blood pressure. They're a standard relationship. I've talked about this before, I think, where some people who are hypertensive are very sensitive to sodium. They eat a couple of potato chips and up goes their blood pressure. Some people eat a bag of potato chips and they gain 2 pounds of weight the next morning, which is due to fluid retention. Okay, so that's the basic relationship between sodium and the ECF volume. And aldosterone will cause increased reabsorption of sodium. And we get the rest of those effects listed. So what are the signals for the release of aldosterone? Well, there are two major ones. One is angiotensinogen 2 from RAAS. And that gets activated by low blood pressure. Assuming we don't have high osmolarity. If it's just a straight loss of blood volume, low blood pressure, the RAAS is activated. Angiotensin 2 targets the adrenal cortex and causes release of aldosterone. The other major signal for the release of aldosterone is hyperkalemia. And the next slide, we're going to look at the details of aldosterone and how it works. So these are the two big ones. Angiotensin 2 and hyperkalemia. I think I said earlier angiotensinogen 2. No, it's angiotensin 2 from RAAS. Okay? And hyperkalemia. And let's make a note in here. High potassium leads to a depolarization of the RMP. We'll come back to that a little bit later, but you should get quite alarmed with potassium imbalances. Remember that there's very little potassium in the interstitial fluid, in the blood plasma, and potassium has a large effect on the resting membrane potential. And I may do a little short lecture on potassium and the RMP. Third signal for the release of aldosterone is significant hyponatremia, if we have very low sodium. And normally that is not a problem with a high sodium, with a typical high sodium diet. Normally not an issue. So the major signals are angiotensin II and hyperkalemia. Next slide looks at the details of aldosterone. So, similar setup, blood vessel on the left, and cells of, oh yeah, do you remember where aldosterone works in the nephron? It worked in, it targets those cells in the distal 1/3 of the DCT and the cortical collecting duct. So later in the nephron. Again, our sodium reabsorption is being regulated by aldosterone. In the proximal convoluted tubule, sodium was not regulated. We were just reabsorbing it as fast as we could. So here comes aldosterone. It's little yellow dots in the blood. It's a steroid, of course. So it just diffuses straight through the cell membrane, simple diffusion, and binds to an aldosterone receptor in the cytoplasm. And those two together then enter the nucleus, and that leads to gene activation. We have transcription, and then we leave the nucleus, the mRNA, and we have translation. We're making new proteins, so those are the new proteins. Remember what we're going to do? We're going to reabsorb sodium. Okay, so what kind of proteins do we make to help reabsorb sodium? Well, here's what's going on with the reabsorption of sodium. Here's the sodium-potassium pump. And remember that it's pumping sodium out. So 3 sodium out, and it's pumping in 2 potassium. Okay. And it's burning ATP. So some of the proteins that get made are new pumps. And then the potassium, so we're speeding up the pumps, we're retaining more potassium. So the potassium then leaves through these facilitated diffusion channels. Facilitated diffusion, I'm abbreviating, channels. So we make new channels and we make new pumps. The other thing that the proteins do, there's a bunch of proteins that get made. Another function of the protein, so let's say new proteins equal pumps and channels. And also, what do they call them here? Do they call them modulator proteins? Yeah, let's just call them modulator proteins. Basically, to speed up existing pumps and channels, make them more efficient. So we're speeding up existing ones and we're inserting new ones. And the end result is that we reabsorb more sodium than we were before. The thing to note, though, is that we're also excreting potassium. So the yellow is the urine, is the waste. The sodium is being reabsorbed from the lumen, from the urine, or what's going to become the urine, back into the blood, but the potassium is leaving. More potassium is flowing out, so we have to watch for that. Okay. So what were the two signals for the release of aldosterone? One was angiotensin II, and what was the other signal for the release of aldosterone? Hyperkalemia. So here is the hyperkalemia, and we're getting rid of the excess potassium and we're pushing it into the urine through the action of aldosterone on these cells in the nephron. Okay. The last slide, so there's a good slide on aldosterone, how it works, sort of its cellular mechanism. And the next slide is a review of RAAS again. This is an important system. If you're heading into the nursing department, they will expect you to remember a fair amount of this. Not all of it, but they expect that when they review it, that it'll come back to you. So let's review it again. So RAAS is all about raising blood pressure. So here's our stress. We'll start with that. Drop in blood pressure. They've put the liver in here, constantly producing plasma proteins, in this case, angiotensinogen. Drop in blood pressure. The granular cells of the kidney produce and release renin. So we looked earlier at several ways of stimulating those granular cells to release renin. So I might say review stimulus for release of renin. So if you're thinking a systemic drop in blood pressure, let's just go back and review before we get too far into this. How are we detecting a drop in blood pressure? What's the first thing that comes to mind? A drop in systemic blood pressure. Who's detecting it? Right, the baroreceptors. And that feeds into the nervous system. And then the sympathetic nervous system, since this is going to be a sympathetic response, fight or flight to raise the blood pressure, The sympathetic nervous system then sent sympathetic neurons to those granular cells, directly to those JG cells, and they release renin. That was one way, probably the major way with a significant drop. So at any rate, you have the circulating angiotensinogen in the plasma. Now renin comes on the scene and converts it to angiotensin 1, which gets converted by ACE into angiotensin 2. And then there are a series of targets again for angiotensin 2. And we looked at this. You have this in an earlier set of notes. But it's good to review again. Maybe make flashcards until you have those targets down, and then you can retire the flashcards. But we talked about the arterioles and how angiotensin 2 is a strong vasoconstrictor. That, of course, leads to blood pressure increasing. There's the cardiovascular control center again. And you could describe this cardiovascular response. What would you talk about here to raise blood pressure? Well, you could speed up the pump. We could increase cardiac output by increasing the heart rate and the force of contraction, and we could also increase vasoconstriction. Okay. Angiotensin 2, back to the hypothalamus, increased vasopressin, or increased ADH. That's going to retain more water, less of it's lost. That will increase the blood volume, increase the thirst, increases blood volume, and then we get over to the adrenal cortex, release of aldosterone, sodium, etc. So, fix the stress. So I would encourage you to make some simple diagrams. They don't have to be pretty. All you're doing is trying to see if you remember and can explain the basic concepts. Okay. Next lecture is the last one for Module 7. See you soon.